So, um, I want you to imagine for a second that you are standing in the middle of a radioactive exclusion zone.
Okay, that's an intense place to start.
Right. But just picture it. You're wearing this heavy sealed hazamat suit. Your dosimeter is just clicking frantically. And you've been tasked with finding a way to clean up some of the most toxic, heavily contaminated soil on Earth.
Wow. Yeah.
And you look down, and your secret weapon for averting this massive ecological disaster is just pushing its way up to the dead pine needles. And it's a gooey, pitch-black, bizarre-looking mushroom that it happens to taste faintly of zesty lemon.
It's a profoundly jarring image, I know, but it's an entirely real one.
Yeah.
I mean, when we look at the natural world, we inherently trust the categories we've built, you know. We like things to be visible, to be neatly organized into little taxonomic boxes.
Oh, really? Like plant, animal, fungus, good bed.
Exactly. We label their behaviors as mutualistic or parasitic, toxic or healing.
But the deeper you venture into mycology, into that damp, dark, tangled undergrowth of the forest floor, those neat little boxes just completely dissolve.
They absolutely do.
Because as a learner, I know you're always looking for those perfect aha moments, those times where the world suddenly makes a little more sense.
Right.
So today's deep dive is going to deliver one of those moments by basically destroying everything you thought you knew about how nature organizes itself, how organisms interact, and how we might actually save our own environment.
Yeah, because we're looking at an organism today that is essentially an evolutionary trickster. It's a highly sophisticated botanical hacker and a nuclear waste sponge, all rolled into one entirely bizarre, primitive-looking package.
It really is the absolute definition of ecological muddy waters.
It is.
So our mission today is to explore the wildly paradoxical existence of Gomphidius glutinosus.
Also commonly and somewhat affectionately known as the slimy spike.
The slimy spike, or if you prefer a slightly more dramatic title from some of the older field guides, the hideous Gomphidius.
Which is just a great name.
It's incredible. So we're going to unpack the sources you shared with us to reveal how this unassuming, rather gooey mushroom defies its own genetic lineage, how it, pulls off microscopic heists against its subterranean neighbors.
Right.
And surprisingly, how it might hold the biochemical key to cleaning up catastrophic radioactive disasters.
Yeah, it's wild. And to really grasp the strangeness of this organism, I want you to visualize the setting where it thrives.
Okay, paint the picture for us.
So imagine a dark, damp, coniferous forest floor, like maybe a dense stand of spruce trees in the Pacific Northwest or Northern Europe.
Lots of moss, lots of shadows.
Exactly. The air is heavy, it smells like decaying pine needles and wet earth, and pushing through that acidic soil is a mushroom that looks exactly like a heavy, slimy iron spike being driven upward from the ground.
Like it was hammered in from below.
Right, yeah. And it's coated in this remarkably thick, gelatinous slime. I mean, it looks prehistoric. To a trained eye, it just looks fundamentally out of place.
And looking out of place is exactly where we need to start. Because before we can even begin to understand its environmental superpowers, like its ability to suck up radiation, we have to look at its profound identity crisis.
Oh, its identity crisis is legendary.
Right, because even the very first scientists who stumbled across this thing in the woods and tried to catalog it were just completely fooled by its disguise.
They absolutely were. And to understand why they were fooled, we have to kind of step back into the scientific landscape of the 18th century.
Yep.
In 1774, there was this German polymath and clergyman named Jacob Christian Schaefer, and he first formally described this mushroom. He called it Agaricus glutinosus.
Hyper-Accumulation.
Right. Now, we have to remember what mycology was like in the 1700s. It was heavily reliant on macroscopic features. I mean, they didn't have DNA sequencing.
Barely had functional microscopes, probably.
Exactly. They barely had microscopes. They categorized fungi based almost entirely on what they could see with the naked eye while literally just standing in the woods.
So if it looked like an apple, they put it in the apple box.
Basically, yeah. And in those days, the genus Agaricus was essentially this giant, messy catch-all category for pretty much any mushroom that had gills.
Right, gills.
Yeah. So if you pick a common button mushroom from the grocery store and look underneath the cap, you see those thin, paper-like ridges radiating outward from the stem. Those are gills.
Like the classic mushroom look.
Exactly. So to early mycologists like Schaefer, gills meant Agaricus. And since the slimy spike had these very thick, pronounced gills running down its stem, he classified it as such. He saw gills, he applied the label, and he moved on.
Done and dusted. But, science doesn't stay still. As microscopes improved and mycologists started looking closer at the actual cellular structure of these organisms, they realized that just having gills didn't necessarily mean two mushrooms were closely related,
Not at all. And that realization led to a massive taxonomic reckoning.
like a total reshuffling of the deck.
Right. So fast forward to 1838, and a Swedish botanist named Elias Magnus Fries, who, by the way, is widely considered the founding father of modern fungal taxonomy, he took a much closer look at Schaefer's Agaricus glutinosus.
And what did he see?
Well, Fries recognized that this slimy, heavy mushroom was just profoundly different from the standard, delicate, gilled mushrooms it had been grouped with. He realized it needed its own distinct category, so he moved it into an entirely new genus, which he named Gomphidius.
Which is such a brilliant descriptive name.
Mm-hmm.
Let's talk about the etymology for a second, because the names scientists choose often tell you exactly what they thought was most striking about the organism.
Mm-hmm.
Gomphos is a Greek root, correct?
It is, yeah. Gomphos roughly translates to plug or large wedge-shaped nail.
Wow.
Right. It was often used to describe those massive conical wooden bolts or iron spikes they used in ancient shipbuilding to hold heavy timbers together.
A spike.
Exactly. When you look at a young, immature, slimy spike, the name is perfectly apt. It has this tapering, heavy stem and a rounded, incredibly dense cap. It genuinely looks like a structural spike forged by nature.
And the specific epithet, glutinosus, is simply direct Latin for sticky or slimy.
Yep, the sticky spike.
The sticky spike. But here's the grand paradox of this mushroom. Schaefer saw gills, so he grouped it with the gilled mushrooms. Fries saw it was different and gave it a new name. But our sources highlight that modern science, like using highly precise molecular DNA sequencing and chemical analysis, tells a completely contradictory story that throws both of those early observations into total chaos.
Chaos is honestly the best word for it, because when modern mycologists sequence the DNA of the slimy spike, and when they analyze its internal biochemical pathways, they find that it doesn't belong anywhere near the agarics.
Wait, really?
Yeah, it has absolutely no direct evolutionary relationship to the vast majority of gilled mushrooms. Instead, its DNA places it firmly inside the order Bulltails.
Okay, I have to stop you there, because for anyone who has ever been foraging or even just watched a documentary on mushrooms, the bulltails, like the bolete family, they are famous for one specific defining trait. They don't have gills.
They absolutely do not. Think of the classic porcini mushroom, the king bolete. Chefs all over the world prize it. If you take a porcini and look under the cap, you don't see paper-thin ridges. You see a spongy, dense mass of microscopic tubes or pores.
Yeah, it looks like a kitchen sponge.
Exactly. Dropping spores out of a sponge-like matrix is the defining macroscopic characteristic of the entire bolete lineage.
Yet here is Gomphidius glutinosus, sitting genetically dead center in the bolete family tree, just stubbornly sporting a thick, waxy set of gills that run deeply down its stem.
Right, what mycologists call decurrent gills.
So it has completely abandoned the signature look of its own ancestors. It has the DNA of a sponge, but it built itself a set of gills. Why on earth would an organism do that? I mean, why spend massive amounts of evolutionary energy to evolve gills if your entire genetic lineage has been highly successful using pores for millions of years?
It's actually one of the most elegant examples of a biological concept called evolutionary convergence.
Convergence, okay.
Yeah, convergence happens when two completely unrelated organisms evolve remarkably similar physical traits. Not because they share a recent ancestor, but because they're trying to solve the exact same environmental problem.
Okay, right. So like the classic textbook example is birds and bats.
Perfect example.
They're from completely divergent lineages. One is avian, evolving from dinosaurs. The other is mammalian.
Hmm.
But they both face the environmental opportunity of the open air, and so both lineages independently evolve wings to solve the problem of flight.
Exactly. Or how sharks and dolphins both evolved dorsal fins and streamlined torpedo shapes, despite one being a fish and the other being a mammal that returned to the sea. The water itself forces them into that shape.
Right. So the environment is the ultimate sculptor.
Precisely. And in the case of our mushroom, the bulltails order and the true gilled mushrooms of the agaricales order evolved gills completely independently of one another. The ancestors of the slimy spike definitely had pores, but over vast stretches of time, the specific environment it lived in exerted a selective pressure that made a sponge a distinct liability.
Wow.
And it made gills a significantly better option for survival.
Okay, so what kind of environment pushes a fungus so hard that it literally ditches its ancestral spongy pores and completely re-engineers its reproductive hardware?
To answer that, you have to look at its preferred habitat. This mushroom loves very specific, challenging environments. It thrives in wet, cold, coniferous habitats. It forms these very close relationships with spruce trees in northern climates and high-altitude mountainous regions.
Okay, so cold and wet.
Very cold, very wet. Now, imagine a spongy pore surface in a constantly freezing, dripping, saturated environment. A sponge is designed to hold water.
Oh, I see where this is going.
Right. If that pore surface becomes waterlogged, the microscopic tubes swell shut. The mushroom can't drop its spores into the air currents. Furthermore, a waterlogged sponge in a freezing environment turns to ice.
Which destroys the cells.
Exactly. It destroys the cellular structure, or it becomes a breeding ground for bacterial rot.
So the sponge just becomes a deadly trap.
It does. Gills, on the other hand, provide a massive amount of surface area for spore production, but their vertical, spaced-out structure allows excess moisture to shed efficiently, much like a shingled roof sheds rain.
Oh, that's brilliant.
And furthermore, the gills of the slimy spike are notably thick and waxy, and when the mushroom is young, those gills are heavily protected by that universal glutinous slime veil. It is an elegant, highly specialized evolutionary solution to the problem of reproducing in a freezing wet spruce forest.
Okay, let's actually look a bit closer at that disguise. Because the sources describe the physical morphology, the shape and structure, in vivid detail. They say the cap ranges from 5 to 12 centimeters across. It starts out almost perfectly spherical, like a hemisphere, and then flattens out as it matures.
Often leaving a little bump, a central boss, right in the middle, yeah.
Right. And covering that entire structure is the slime.
The slime is incredible. We aren't just talking about a mushroom that feels a bit damp to the touch. It is coated in a universal glutinous veil.
A universal veil.
Yes, it's this thick, transparent, highly viscous gelatinous layer that encases the entire fruiting body when it first emerges from the soil. As the cap expands outward to open its gills, that gelatinous layer stretches tighter and tighter until it eventually tears.
And when it tears, it leaves a sticky remnant on the stem, right?
Yes, the tearing leaves a slimy ring or an annular zone around the upper part of the stem. And here is where the color palette of this mushroom gets really interesting.
Because it's pretty drab at first, right?
Yeah, the cap itself is usually a dull, grayish purple or a muted mauve. But as the mushroom matures, it begins dropping its spores. And the spores of Gomphidius glutinosus are incredibly dark. They're a deep sepia brown, almost bordering on black.
Okay.
As these millions of dark spores fall from the gills, they get caught in that sticky slime ring on the stem, turning the ring and the gills themselves a bruised, pitch black color.
Which is where the hideous moniker comes from.
Exactly.
But the very bottom of the stem is completely different.
The contrast is startling. While the top half is gray, purple, and blackening slime, the base of the stipe, the bottom of the stem where it meets the soil, is a vibrant, deep, unmistakable chrome yellow.
Like highlighter yellow.
Yes. And it's not a superficial dusting of color either.
Yeah.
If you take a knife and cut the stem in half longitudinally, you'll see that bright, acidic yellow color runs deep through the dense flesh of the base.
It's just an incredibly weird, visually contradictory organism.
It really is.
But you mentioned earlier that modern science proved it was a bolete. Does that disguise hold up if you put it under a microscope? Like if you actually look at the cells?
Not at all. The naked eye sees an agaric, but the microscope immediately exposes the bolete lineage.
How so?
If you take a razor blade, slice an unimaginably thin cross-section of those waxy gills, and put it under a high-powered lens, the illusion just shatters. The microscopic features scream bull tails.
Give me an example.
For instance, the spores themselves are massive. They are described as subfusiform.
What does subfusiform mean in practical terms?
So fusiform means shaped like a spindle, like wider in the middle and tapering gracefully to a point at both ends, kind of like a tiny football. Subfusiform means they are somewhat spindle-shaped, perhaps a bit asymmetrical.
Okay, got it.
But it's their size that is remarkable. They measure between 15 and 21 micrometers in length.
Is that big for a spore?
Oh, in the microscopic world of fungal spores, that is absolutely gargantuan. Furthermore, the surface of the gills is covered in specialized sterile cells called cystidia. Yeah. In the slimy spike, these cystidia are so large, so prominent, and so numerous that they actually give the surface of the gills a hairy texture when viewed under magnification.
Wow.
And these massive spores and prominent cystidia are classic textbook hallmarks of the bolete family.
So we have an organism that is fundamentally a structural paradox.
Hmm.
It went to immense evolutionary trouble to look and function like one thing while genetically remaining something else entirely.
Exactly.
But as we dig into the sources, we discover that looking like an imposter isn't just a quirky physical trait. It actually mirrors its entire lifestyle.
Oh, absolutely.
Because to understand why this mushroom evolved such highly specialized extreme traits, we have to look at how it gets its food, and more importantly, who it robs to survive.
Right. When we pull back the lens and look at the broader forest ecology, we enter the hidden world of subterranean fungal networks. For decades, the established scientific consensus was that Gomphidius glutinosus was just a standard, friendly ectomycorrhizal fungus.
Okay, let's break down that term, ectomycorrhizal, because it's foundational to understanding how forests actually work.
It is the biological engine of the forest. Ecto means outside, myco refers to fungus, and rhiza means root. Ectomycorrhizal fungi are organisms that send their microscopic threads, their mycelium, through the soil and wrap them intimately around the outside of the root tips of living trees. They form a deeply mutualistic partnership.
Like a trade agreement.
Precisely. The tree stands above ground, capturing sunlight and using photosynthesis to manufacture carbon-heavy sugars. The fungus lives underground, unable to photosynthesize. So the tree feeds the fungus these sugars.
Okay. And what does the tree get out of it?
In return, the fungal mycelium acts like a massive, highly efficient secondary root system for the tree. The incredible fine fungal threads can penetrate micropores in the soil that tree roots simply cannot reach. They scavenge for water, nitrogen, phosphorus, and trace minerals, and they trade those vital elements back to the tree.
This is what popular science often calls the wood wide web.
Exactly, yes.
It's a cooperative economy. The tree gets the minerals it needs to grow, the mushroom gets the sugar it needs to survive. And for a very long time, mycologists just assumed the slimy spike was just a good, upstanding citizen of this economy, playing by the rules and specifically partnering with spruce trees.
And honestly, it made perfect sense based on observation. I mean, you always find them growing under spruce. But modern science, utilizing isotopic tracing, genetic sequencing, and incredibly precise electron microscopy, revealed a much darker, much more fascinating reality beneath the soil.
Oh, I love where this is going.
Gomphidius glutinosus is not a simple, friendly mutualist. It is a hypersymbiont. In more direct terms, it is a biotrophic mycoparasite.
Meaning?
It is a fungus that actively parasitizes other fungi.
So it's not trading with the tree directly at all. It's hacking the existing system.
Exactly. It specifically tracks down and targets the mycelial network of a completely different genus of mushroom, the Suillus mushrooms.
Suillus, okay.
Think of it this way. A Suillus mushroom has spent immense biological energy building a beautiful, mutually beneficial relationship with a massive spruce tree. The tree is pumping vast amounts of carbohydrates down into the roots. The Suillus is industriously pumping soil minerals up into the tree.
Like an underground fiber optic network, passing vital data back and forth.
And then the slimy spike moves into the neighborhood. It doesn't approach the tree to offer a trade. It acts as a biological wiretap.
A wiretap?
Yeah, it physically tracks the chemical signature of the Suillus mycelium through the soil. When it finds it, it taps directly into the Suillus network, siphoning off the massive flow of nutrients that the Suillus worked so hard to establish.
That's insidious.
It is a tripartite, or three-way relationship. The spruce tree manufactures the carbs, the Suillus trades for those carbs, and the Gomphidius taps into the Suillus and just steals everything.
Wait, let me make sure I'm understanding the mechanics of this theft. It's not giving anything back to the tree.
No.
It is literally just a hacker tapping into the forest internet, stealing the bandwidth, and offering zero services in return.
It offers absolutely nothing to the tree or the host fungus. And the cellular mechanics of how it pulls off this microscopic heist are just staggering.
How does it do it?
To tap the wire, the Gomphidius uses highly specialized infection structures called haustoria.
Haustoria.
Yeah, think of a haustorium as a microscopic biological syringe. The Gomphidius hyphae, its searching fungal threads, grow alongside the Suillus threads in the dark soil. When they find the perfect vulnerability, usually right at the exact junction where the Suillus has penetrated the outer cortical cells of the tree's root, the Gomphidius deploys this haustorial syringe. It pierces through the tough outer cell wall of the host Suillus fungus.
But it doesn't kill it, because the term you used earlier was biotrophic parasite. Bio meaning life. It needs the host alive.
And that is the genius of it. If it simply killed the Suillus cell, the flow of nutrients from the tree would instantly stop, and the Gomphidius would starve. It has to keep the host alive and functioning while draining it.
Right.
So the haustorium pierces the rigid outer cell wall, but it stops just short of piercing the delicate inner cell membrane. It pushes against that membrane like a finger pushing into a balloon, wrapping itself in the host's membrane without popping it.
Oh, wow.
This creates an incredibly intimate, microscopic interface between the thief and the victim.
Okay, so it has breached the vault, but how does it actually get the gold out? Because normally in biology, nutrients want to flow down a concentration gradient from where there is a high concentration to where there is a low concentration.
Right.
If the Gomphidius is stealing all this food, it must be packed full of nutrients itself. So how does it force even more food inside?
Right. It has to pull those nutrients across the boundary against the natural flow of physics. To do that, the Gomphidius builds what is effectively a localized cellular battery.
A battery. Really?
Right. Yes. It uses a specialized enzyme embedded in its membrane called an H+ATPase pump. What this molecular machine does is aggressively pump protons, positively charged hydrogen ions, out of its own cell and into the infinitesimally small gap between itself and the Suillus membrane.
So it's forcibly ejecting protons to create an imbalance.
Yes, it is constantly pumping protons out, creating a massive, localized concentration of positive charge in that tiny gap. Think of it exactly like pumping millions of gallons of water up into a reservoir behind a massive hydroelectric dam. You are fighting gravity to put the water up there, but once it's there, you have created immense potential energy.
So you have this huge buildup of protons on the outside.
Yeah.
And the laws of physics dictate that they are absolutely desperate to flow back inside to balance the electrical and chemical pressure.
Precisely. And the Gomphidius takes advantage of that desperation. It provides specialized gated doors in its membrane for those protons to flow back through. These doors are called symport carriers. But the door has a very specific locking mechanism. It only opens if the rushing proton brings a stolen nutrient molecule with it.
Oh, that is devious.
It's a marvel of evolutionary engineering. For stealing carbohydrates, the sugars from the tree, it uses a specific symport carrier called HXT1. For stealing amino acids, the building blocks of proteins, it uses a suite of carriers called AAT.
So it's very targeted.
Highly targeted. As the protons violently rush back into the Gomphidius cell to relieve the pressure gradient, they physically drag the stolen sugars and amino acids from the Suillus through the door with them. It is highly active, biochemically powered theft occurring at a molecular scale millions of times a second.
I am genuinely mind-blown by the sheer mechanical elegance of that system.
It's beautiful in a dark sort of way.
But knowing how aggressively it operates, I have to ask a bigger ecological question. If it's building cellular batteries to actively steal all this energy, doesn't this severely damage the spruce tree, or at least kill the Suillus network? The sources we have make a surprisingly counterintuitive note that this intense parasitism might not actually harm the host system in a significant way. How is that even possible?
It's a great question, and it forces us to reconsider how we define harm in complex ecological networks. In a human context, someone tapping your bank account and stealing your income is universally harmful. It destroys your stability.
Right.
But a forest ecosystem doesn't operate like a single bank account. The dynamics are fluid and incredibly resilient. A single Suillus mycelial network is vast. It can span huge areas of the forest floor, connecting with multiple trees.
It's highly decentralized.
Exactly. The Gomphidius isn't attacking the entire grid, it is tapping into highly localized nodes. Many forward-thinking ecologists speculate that the presence of a localized parasite might actually act as a stimulant for the broader network.
A stimulant, how so?
By creating a localized nutrient sink, a void where food keeps disappearing, the Gomphidius might inadvertently trigger the Suillus network to forage more aggressively, to expand its reach further through the soil, and to increase its trading volume with the spruce tree to make up for the deficit.
So, like stressing a muscle during a workout causes micro tears that ultimately force the muscle to grow back stronger and more capable.
In a metabolic sense, yes, exactly. The Gomphidius is walking a microscopic tightrope. It relies entirely on the overall health and vigor of both the Suillus network and the tree canopy above. If it gets greedy, if it drains the local nodes to the point of systemic collapse, the host dies and the parasite starves to death right alongside it.
Mutually assured destruction.
Precisely. So, over millions of years, it has evolved to exist in this perfect, tense equilibrium skimming, just enough high-value resources off the top to fuel the rapid growth of its massive, fleshy, desperately slimy, fruiting bodies above ground, without collapse. It's the underlying economy of the soil that supports it.
That balance is just brilliant. But pulling off a microscopic heist of that caliber, infiltrating another living organism, breaching its defenses, actively pumping out its lifeblood, and defending your own body while you do it, that requires a very specialized, very dangerous toolkit.
It certainly does.
And for Gomphidius glutinosus, as our sources detail heavily, that toolkit isn't just physical haustoria. It is made of entirely unique, bespoke chemistry.
Right, because you don't become a highly successful, specialized parasite by blending in biochemically with your surroundings. You have to synthesize compounds that protect your vulnerable tissue, facilitate your theft, and aggressively ward off any competitors who might want to steal what you've already stolen.
Makes sense.
Gomphidius glutinosus is a master chemist. Like many fungi, it relies heavily on a metabolic engine called the shikimate-corismate pathway.
Okay, let's slow down and define that. The shikimate-corismate pathway, that sounds incredibly dense. What exactly is that doing inside the mushroom?
Think of a metabolic pathway as a biological factory assembly line. A living fungus takes in basic, simple molecules, things like sugars and simple amino acids. These are primary metabolites, the basic fuel needed just to keep the cell alive.
Okay.
But the fungus takes some of these basic materials and diverts them onto this specific shikimate-corismate assembly line. Through a series of highly orchestrated enzymatic reactions, it transforms those basic materials into highly complex, specialized molecules called secondary metabolites.
Okay, so primary metabolites are like the steel and rubber, the raw materials, and secondary metabolites are when the factory turns that steel and rubber into an armored tank. They aren't strictly necessary for the basic functions of life, like breathing or dividing, but they give the organism a massive, specialized competitive edge in a hostile environment.
That is a perfect analogy, yes. Now, within the entire Bulltails order, the massive family tree that the slimy spike belongs to, the most famous and common product rolling off this metabolic assembly line is a compound called xerochromic acid.
Oh, right. If you've ever been walking in the woods, picked up a beautiful bolete mushroom, pinched the yellow spongy underside with your thumb, and watched it instantly, almost magically, turn a vivid bruised blue color, you are watching xerochromic acid oxidize upon contact with the air.
Exactly.
It's a rapid chemical defense mechanism to deter insects from eating it.
It is, and it's a classic bolete trait. But Gomphidius glutinosus, despite being genetically a bolete, does not manufacture xerochromic acid. Its assembly line has been entirely retooled.
Because, of course, it has to be different in every conceivable way. It rejects the family tradition.
It exclusively produces its own distinct signature compound. Gomphidic acid.
Gomphidic acid.
Yes. This is a highly specialized pulvinic acid derivative. And it is so unique, so incredibly specific to this exact mushroom that mycologists use it as a chemotaxonomic fingerprint.
It's like a barcode.
Exactly. If you analyze a tiny sample of unidentified fungal tissue and find a gomphidic acid, you don't even need to look at the DNA. You know with absolute certainty you are looking at the slimy spike.
So gomphidic acid is its chemical signature. But what does this bespoke molecule actually do? What is the function of this chemical armor?
Well, it's the ultimate biochemical multitasker. It serves several crucial biological functions simultaneously. First, these pulvinic acid pigments act as incredibly powerful natural UV screens. They absorb high-energy ultraviolet radiation from the sun, dissipating it before it can penetrate the fungal cells and shatter the delicate DNA inside.
Wait, I have to push back on that logic for a second. We set the stage earlier by placing this mushroom in a dark, damp, heavily shaded coniferous forest. It grows under the dense canopy of spruce trees. Why on earth does a mushroom growing in the shadows need a high-powered, bespoke chemical sunscreen? It's not sunbathing on a beach.
It's a very astute question. While it's true they are deeply associated with shaded spruce forests, they are not strictly confined to the dark valleys. Our sources explicitly note that Gomphidius glutinosus can thrive in high-altitude alpine environments, growing alongside scrubby spruce lines all the way up to 2,900 meters.
Wow, that's over 9,500 feet above sea level.
Exactly. At those extreme elevations, the tree cover becomes sparse, the atmosphere is considerably thinner, and the intensity of the UV radiation striking the ground is exponentially higher.
Oh, I see.
Without a robust chemical shield woven into its flesh, a fleshy mushroom exposed at 9,000 feet would suffer catastrophic DNA damage and cellular death very quickly. Gomphidic acid allows it to colonize environments that would fry less chemically sophisticated fungi.
That makes perfect sense. So extreme UV protection is function number one. What else does this acid do?
Function number two is localized defense. The forest floor is not a peaceful place. It is teeming with millions of hungry bacteria, predatory nematodes, and insect larvae that would love nothing more than to feast on a massive, nutrient-rich, fruiting body full of stolen carbohydrates.
Right, it's a buffet.
These fulvinic acid derivatives possess potent antimicrobial and anti-fedin properties. They make the tissue highly unpalatable and in some cases chemically toxic to these micro-competitors. It's a chemical fence keeping the scavengers away from the stolen loot.
Okay, UV shield, bug repellent. What's the third function?
Function number 3 is the one that sets the stage for the most staggering, almost unbelievable part of this organism's story.
Okay, I'm ready.
The chemical structure of Gomphidic acid, specifically the presence of multiple carboxyl groups, makes it an exceptionally powerful chelating agent.
Chelation. I've heard that term in medicine regarding heavy metal poisoning. It means binding to metals, right? Like microscopic molecular claws that grab onto stray metal atoms in the environment and lock them up.
That is exactly what it is. The carboxyl groups act like molecular pincers. As the fungal network grows through the soil, it inevitably encounters naturally occurring heavy metals, things like lead, copper, or zinc. In high concentrations, these metals can interfere with the fungus's cellular machinery. The Gomphidic acid complexes with these metal ions, grabbing them and locking them into stable, inert structures so they can't cause biological damage.
And what's fascinating is that as a mycologist out in the field, you can actually see this complex chemistry happen in real time. The sources describe a very specific chemical test used for field identification involving Meltzer's reagent.
Yes, the chemistry is highly reactive and visible. If you are foraging and you find a slimy mushroom and want to definitively prove it is Gomphidius glutinosus, you look at that vibrant chrome yellow flesh at the very base of the stem. You apply a single drop of Meltzer's reagent, which is a specialized iodine-based solution.
And what happens?
Because of the highly specific amyloid and dextrinoid compounds present in that Gomphidic acid-rich yellow tissue, the reaction is dramatic and immediate. The bright yellow flesh instantly stains a dark, bruised green, eventually turning almost pitch black.
And there's a different test for the slime on the cap, right?
Yes. Conversely, if you take a strong base, like a 3% solution of potassium hydroxide, or KOH, and apply it to the slimy, gelatinous cuticle of the cap, the mass of cells won't turn black. They will take on a very distinct pale bluish cast. These rapid color changes aren't magic. They are visual proof of the complex, highly specific secondary metabolites interacting with the chemical reagents.
And it's not just mycologists doing cardi tricks in the woods. Researchers are actually looking deeply into harvesting these specific basidiomycete pigments for our own use.
Right. Absolutely.
Because if an organism has evolved a chemical that is stable enough to act as an industrial-grade UV shield at 9,000 feet and toxic enough to repel insects without killing the soil, that could be incredibly useful for human engineering.
It is a rapidly expanding frontier in industrial biochemistry. Currently, human manufacturing relies heavily on synthetic chemicals for industrial dyes, food colorants, and UV stabilizers. Many of those are derived from petroleum and have highly questionable long-term environmental and human health impacts.
Right.
The fungal pigments produced by the slimy spike, however, are highly stable under extreme heat and light. They bond strongly to various substrates, and most importantly, they are entirely biodegradable. They represent a sustainable, eco-friendly alternative for the future of industrial chemistry.
That's amazing. So binding to trace heavy metals chelation is a really neat evolutionary trick when you are a mushroom growing quietly in a pristine, ancient spruce forest. It's a great defense mechanism.
It is.
But as we know, the world is no longer pristine. Here is where the story of the slimy spike takes a sharp, incredibly dark turn. Because when human beings introduce massive industrial pollution, or worse, catastrophic nuclear disasters into the environment, this neat chemical survival tool goes from being a biological curiosity to a terrifying and awe-inspiring side effect.
This brings us to the grim legacy of April 1986.
Chernobyl.
Right, the catastrophic meltdown of reactor number 4 at the Chernobyl nuclear power plant in Ukraine. When that reactor exploded, it didn't just contaminate the immediate facility, it ejected a massive, invisible plume of highly radioactive isotopes high into the atmosphere, which weather patterns then dragged across massive swaths of Eastern and Western Europe.
And as the rain fell, it washed these isotopes down into the soil of forests thousands of miles away.
Precisely. And the most pervasive, dangerous, and long-lasting of those isotopes was cesium-137.
Right, cesium-137.
Exactly. Cesium-137 is a highly radioactive byproduct of nuclear fission. It emits powerful gamma radiation and has a half-life of about 30 years, meaning it persists in the environment for over a century, continuously irradiating everything around it.
A total nightmare for the ecosystem.
In the years following the disaster, radioecologists went into the exclusion zone, as well as the heavily affected forests in Germany, Poland, and Scandinavia, to measure the ecological fallout. They expected to find dead forests.
But they didn't.
Instead, they made a shocking discovery regarding the fungal kingdom. Certain types of mushrooms weren't just surviving the intense radiation, they were acting as aggressive biological radiological sponges.
And sitting right at the absolute top of the list of these nuclear sponges was our strange, gooey friend, Gomphidius glutinosus.
It is classified as a hyperaccumulator. It absorbs and concentrates cesium-137 at levels that completely defy logic and standard biological models.
How much are we talking about here?
To give you some context on the scale of this absorption, a standard plant might take up a trace amount of a mineral from the soil. The slimy spike can concentrate radioactive cesium at levels exceeding 10,000 times the background concentration of the soil it is actively growing in.
10,000 times.
Yeah.
That's not passive absorption. The mushroom isn't just sitting there soaking up what washes over it. It is actively, mechanically, vacuuming the radiation out of the dirt. Let's look at the specific data from the sources, because the numbers are almost unbelievable.
The numbers are staggering. In a landmark study conducted in the Chernobyl area in 1993, researchers tested the fruiting bodies of Gomphidius glutinosus growing in the highly contaminated soil. They recorded a concentration of 17,117,000 becquerels per kilogram of dry weight of cesium-137.
17 million becquerels.
Yeah.
Just to put that in perspective for you, a becquerel is a unit of radioactivity representing one nucleus decaying per second. Millions of decays per second inside a single kilogram of dried mushroom tissue. That is a violently radioactive object.
It is intensely radioactive. And what is crucial to understand is that this was not a localized, short-term anomaly that faded away after a few years. Because of the long half-life of cesium-137, and because of the specific ecological mechanics of how fungi interact with the soil layers, this hyperaccumulation persists today.
Decades later.
Even decades after the disaster. If you go to specific forest regions in Bavaria, Germany, or in southern Poland and you test the mushrooms, the slimy spike consistently shows massive, off-the-charts spikes in radioactivity compared to the surrounding flora and the soil itself.
But the fundamental question is why? I mean, I understand it has chelating chemicals, but why does it suck up cesium so aggressively? A mushroom has no biological use for a highly radioactive isotope created in a human fission reactor. What does the mushroom think it's doing?
It doesn't want the cesium at all. It is the victim of a very specific, highly efficient case of mistaken chemical identity.
Mistaken identity.
To understand this error, we have to look at the structure of the periodic table of elements. Cesium is classified as an alkali metal. If you look at where it sits on the table, it is in the exact same vertical column as potassium. Because they share a very similar outer electron configuration, their physical size and the way they behave in chemical reactions are remarkably similar.
And potassium is a big deal for mushrooms, right?
Potassium is absolutely vital. Fungi are desperate for potassium. Unlike plants, which use cellulose to give their cells rigid structure, fungi rely on turgor water pressure inside the cells to push their way through hard soil and push a massive fruiting body up into the air.
Okay, that makes sense.
They pump massive amounts of potassium ions through their mycelial networks to regulate the cellular volume, maintain precise water pressure, and balance their internal cellular pH.
Oh, wow. Okay, the pieces are falling together. So the fungus is desperately hunting for potassium to build its water pressure. And remember what we discussed in incredible detail earlier. The slimy spike is a biotrophic parasite. It isn't just casually soaking up water. It has built this massive, hyper-aggressive, high-powered cellular battery, that proton pump we talked about, to violently strip mine nutrients from a vast underground network of Suillus mushrooms.
Precisely. You have an organism that is biologically supercharged to suck nutrients out of a network that spans huge swaths of the forest floor. The Suillus network reaches out, absorbing everything it can find. Because cesium-137 mimics potassium so perfectly, the Suillus accidentally pulls the radioactive isotope into its network, thinking it is gathering essential potassium for its tree partner.
And then the Gomphidius just robs them.
Then the Gomphidius hacks into the Suillus. Its hyper-aggressive parasitic machinery blindly and powerfully vacuums up everything in the pipe. It inadvertently vacuums up the radioactive fallout.
Incredible.
It is effectively drawing from a massive surface area of the contaminated forest floor, filtering it all through the Suillus network, and intensely concentrating all those heavy, mistaken radioactive ions directly into its own fleshy, above-ground body.
And we know exactly where in that fleshy body the radiation goes, thanks to some brilliant, highly detailed radioecology research. There was a specific Polish study mentioned in our sources that didn't just measure the whole mushroom, they painstakingly mapped the vertical transport profile of the cesium-137 as it moved from the earth into the fungus.
Yes, and the distribution map they created is incredibly telling regarding how the fungus operates. They took core samples of the deep soil, the shallow topsoil, the stem of the mushroom, and the cap, and measured the radioactivity of each distinct layer.
What did they find?
The deep mineral soil had the lowest concentration of cesium. The top layer of the soil, the organic horizon where the pine needles are decomposing and where the vast majority of the Suillus mycelial networks are actively foraging, had a higher concentration.
Which makes sense as the fallout landed on the surface and bound to the organic matter.
Right, but the mushroom itself entirely dwarfed the soil levels. It wasn't just matching the soil, it was multiplying it. And within the anatomical structure of the mushroom itself, the distribution was not even.
Where was it concentrated?
The stem of the mushroom averaged significantly higher concentrations of radiation than the fleshy cap above it. In their specific sample data, the tissue of the stem averaged 565.2 becquerels per kilogram, while the tissue of the cap averaged only 322.7.
Because the stem isn't just a physical support, it's a biological highway. It is the bottleneck.
Exactly the right way to think about it. The stipe acts as the primary forced transport conduit. It is the highly pressurized funnel where all the resources gathered by the vast underground network over square meters of soil are forced tightly upward to quickly inflate the cap and produce the spores.
So it all gets jammed in there.
The heavy cesium ions get trapped, they accumulate and embed themselves in the dense tissue of the lower stem, which ironically is that exact same bright yellow Gomphidic acid-rich tissue we discussed earlier. The acid acts as the claw, the stem acts as the funnel, and the radiation is trapped.
So when you put all of this together, we have an organism that acts like a highly efficient, highly localized biological vacuum cleaner for catastrophic nuclear waste. It pulls it from the wide soil and concentrates it into a tiny physical package.
It's astounding.
And researchers and environmental engineers aren't just observing this, they are actually trying to weaponize this biological quirk to clean up the environment, right?
Our sources describe a fascinating theoretical remediation concept called the Japanese landscape protocol.
Yes, it is a profoundly ingenious, if somewhat apocalyptic, application of targeted ecology. It falls under the broader science of mycoremediation, using fungi to heal damaged ecosystems.
How does it work?
The Japanese landscape protocol outlines a theoretical, multi-generational method for reclaiming heavily contaminated land, perhaps surrounding a meltdown site. First, you establish a heavily fenced-off, highly secured nuclear recovery zone. You go in and mulch the area heavily with wood debris, branches, and organic matter to create a rich, deep organic litter layer on top of the radioactive soil.
You are building a massive fungal buffet. Then what? Then you plant native coniferous trees, specifically species like spruce, across the contaminated zone.
The hosts. And then you introduce the fungal internet.
You inoculate the soil with specific strains of Syillus fungi to partner with the spruce trees. You establish the mutualistic network. And then, as the final step, you introduce Gomphidius glutinosus to aggressively parasitize the Suillus.
And then you just wait.
And then you leave. You let the forest grow. The mushrooms do what millions of years of evolution have programmed them to do. They scour the soil, mistaking cesium for potassium, and aggressively draw the radioactive isotopes up from the wide ground and concentrate them into their dense, fleshy, fruiting bodies above ground.
Okay, but then what? You haven't destroyed the radiation? You've just moved it.
Yeah.
Now you just have a spooky forest full of highly radioactive mushrooms glowing in the dark. How does that help clean the site?
Because it is vastly easier to harvest a mushroom than it is to dig up and process 1 million tons of topsoil. Once the mushrooms fruit and concentrate the cesium, you send in specialized teams wearing full hazmat radiation suits. They meticulously harvest the mushrooms from the forest floor before they have a chance to rot and return the heavy isotopes back to the topsoil.
So they pick the radioactive crop.
Yes.
Ugh.
And these harvested mushrooms, which have now successfully concentrated the radioactivity from a vast volume of scattered soil into a relatively tiny amount of biological mass, are transported to specialized, highly controlled facilities. There, the mushrooms are incinerated.
Burned.
Exactly. The water and organic carbon burn off, leaving behind a highly radioactive ash. The volume of this ash is an infinitesimally small fraction of the volume of the original contaminated soil.
You've concentrated the problem into a manageable size.
Exactly. Finally, that highly radioactive ash is subjected to a process called vitrification. It is essentially mixed with glass-forming chemicals and melted down into solid, stable, impenetrable blocks of glass. Those glass blocks, locking the radioactive cesium securely inside, are then transported to deep geological repositories for long-term safe storage. You have successfully used biology to concentrate an unmanageable, diffuse environmental disaster into a solid, manageable package.
Okay, here is where it gets really interesting to me. From a pure biology standpoint, we are talking about using this organism as a tool to absorb radiation levels that would be entirely catastrophic to a human being. Levels that would mutate the DNA of a mammal or kill a plant outright.
Definitely.
What does all this radiation actually do to the mushroom? Does the slimy spike suffer massive cellular damage? Does it mutate into some giant deformed monster? Or is it literally just a biological vacuum cleaner with a death wish, happily poisoning itself in order to clean the forest floor?
You've just hit on one of the most profound, cutting-edge questions in the field of radioecology. You would logically think that pumping millions of becquerels of ionizing radiation through a delicate cellular membrane would violently shred the fungal DNA, causing widespread mutations, cellular breakdown, and death. And in many organisms, that is exactly what happens.
But not fungi.
Fungi, however, possess remarkable, highly robust DNA repair mechanisms that allow them to endure environmental stresses that would instantly kill other kingdoms of life. But the sources note something even more surprising, something that forces us to completely rethink the boundaries of biology.
What's that?
It was discovered in the heavily irradiated zones after Chernobyl that certain types of fungi, specifically dark, melanin-producing fungi, don't just tolerate the high radiation. Their growth might actually be stimulated by it.
Wait, I want to make sure I'm hearing you correctly. Like radiotrophic fungi. In the same way that a green plant uses sunlight for photosynthesis to create energy, are you saying these fungi use deadly ionizing radiation as a food source?
It is a leading, highly researched hypothesis in mycology right now. Melanin is the exact same dark pigment that tans human skin to protect it from UV light. In these fungi, the melanin is densely packed in the cell walls.
Okay.
The hypothesis is that the melanin polymer can physically absorb high-energy gamma radiation and fundamentally alter its own electron configuration. It acts as a biological transducer, potentially converting that deadly ionizing radiation into usable metabolic energy that the fungus can use to grow.
That is literal science fiction. It's turning fallout into food.
It is incredible. Now, it's important to clarify that Gomphidius glutinosus itself isn't the primary textbook example of radiotropism. That specific title usually goes to certain species of black molds that were famously found actually growing inside the ruins of the Chernobyl reactor core.
Right, thriving in radiation fields that would melt robotics.
Exactly. However, the slimy spike's uncanny ability to thrive, to successfully fruit, and to maintain its complex parasitic lifestyle while accumulating lethal, staggering doses of cesium forces us to rethink how life adapts to apocalyptic environments. It's not just a vacuum cleaner with a death wish, it's a testament to the staggering, almost frightening resilience of the fungal kingdom. When human beings make catastrophic, world-altering mistakes, nature adapts to the new reality with breathtaking speed.
That is genuinely awe-inspiring. It reframes the entire organism. But let's bring this soaring discussion of nuclear physics and biological super-adaptation all the way back down to Earth, to you, the learner.
Good idea.
Let's talk about the practical reality of encountering this thing.
Okay.
Because with all this intense talk of nuclear vacuum cleaners, radioactive ash, and toxic heavy metals, what happens when a casual forager is just walking through a beautiful spruce forest, spots a patch of slimy spikes, and decides they want to take them home and make a nice risotto?
It presents what I like to call the ultimate forager's dilemma, Because beyond the extreme example of radioactive cesium, the hyper-accumulating nature of Gomphidius glutinosus applies universally to highly stable, non-radioactive heavy metals as well.
Oh, right.
Its chemical claws are incredibly efficient at drawing up toxic elements from the soil, including lead, cadmium, silver, and chromium.
The sources mention silver accumulation specifically as a major factor.
Yes. Detailed ecological studies conducted in the Pacific Northwest of North America found that various Gomphidius species can accumulate elemental silver in their tissues at concentrations 20 to 80 times higher than the surrounding soil they are growing in.
80 times, wow.
Now, this makes them absolutely incredible bioindicators for scientists. A researcher can map the historical invisible pollution of a forest, perhaps tracing the fallout from an old forgotten copper smelter, or mapping industrial runoff from a factory that closed down decades ago, simply by harvesting and chemically analyzing the tissue of a single slimy spike.
The mushroom acts as a highly accurate, natural historical archive of human pollution, recording chemical data in its flesh when our own instruments weren't looking.
Exactly.
That's great for science, but that historical archive of industrial waste is currently sitting on my dinner plate.
Exactly the problem. The very biochemical mechanism that successfully protects the living fungus, those complex metallothioneins and pulvinic acids binding tightly to the toxic metal ion so they don't harm the mushroom cells, means that those heavy metals are permanently locked in the fungal tissue. They don't wash out.
So if you eat it,
If you harvest and eat a slimy spike from a forest that has a hidden history of industrial pollution or is near a roadway with historical leaded gas exposure, you are ingesting a highly concentrated bioavailable dose of toxic heavy metals.
So let me summarize the culinary pitch for this mushroom. On one hand, depending on where you pick it, it might be heavily loaded with toxic lead or radioactive silver. On the other hand, it's literally called the hideous Gomphidius. It's completely covered in an extreme thick layer of gelatinous slime, and as soon as it drops spores, its gills turn bruised and black.
Yes.
It sounds substantially less like dinner and more like a cursed, toxic artifact from a fantasy novel. Why does anyone, anywhere, ever eat this thing?
Because human culinary tradition is wildly polarizing and often completely defies logic. In many parts of the Western world, specifically in North American and British field guides, it is firmly labeled as not recommended or simply inedible, strictly based on its revolting slimy texture and its unappetizing appearance.
Fair enough.
But in Scandinavia, in parts of Russia, and across Eastern Europe, it has a long history of being highly prized by traditional foragers. Experienced, knowledgeable foragers know that if you can get past the hideous exterior, the flesh inside is surprisingly meaty, firm, and has a very unique, highly sought-after flavor profile.
The sources describe the flavor in a really unexpected way. They say it has a distinct, zesty lemon finish.
It absolutely does, because the tissue is loaded with those specific and natural acids we discussed, the Gomphidic acid and other related compounds. The flesh has a naturally high acidity. This gives the cooked mushroom a sharp, bright, citrusy tang that is highly unusual and deeply prized in a savory mushroom. It cuts through rich sauces beautifully.
But accessing that flavor requires intense, immediate labor. Preparation is everything.
It really is. You absolutely cannot just casually harvest a fresh slimy spike and toss it into a basket alongside your delicate, beautiful golden chanterelles or pristine porcinus.
Because it ruins the basket, right? Why does it do that?
Because of the infamous blackening phenomenon, the mushroom tissue contains incredibly high concentrations of easily oxidizable compounds. If the mushroom gets slightly crushed, bruised, or simply sits exposed to the open air for too long after being picked, these compounds react with the oxygen and turn the entire mushroom pitch black. More disastrously, if that wet, sticky slime touches the other mushrooms in your basket, it stains them permanently black as well, visually ruining an entire day's harvest.
So what's the workaround?
A knowledgeable, traditional forager will execute a very specific preparation right there in the forest. They carefully peel the thick, slimy pellicle completely off the cap, and they aggressively scrape the gooey ring off the stem immediately after picking it, leaving only the firm, white, dry inner flesh to go into the basket.
Okay, so you've peeled the slime off in the woods, your hands are probably a mess, and you get it home. How do you actually cook it? Because if it's adapted to a freezing, wet environment, it must be full of water. If you try to put it in a dehydrator, it just turns into a miserable black smudge, right?
It is notoriously terrible for drying. It contains a massive percentage of water by volume. To cook it properly and extract that lemon flavor, you must sauté it in a very hot, dry pan, or perhaps with just a tiny sliver of butter, and you must have extreme patience.
Let it cook down.
Exactly. As it hits the heat, the cellular walls break down, and it will release a massive pool of dark liquid into the pan. You have to keep cooking, letting it boil in its own juices until all of that excess water completely evaporates and the mushroom finally begins to sear and brown in the concentrated, highly acidic reduction left behind. Only then does the meaty texture firm up and that zesty lemon flavor really shine.
Assuming, of course, that after all that effort, you've actually correctly identified the mushroom in the first place. Because as a forager, especially when dealing with potential heavy metal accumulators and slime, you really don't want to get the identification wrong. Our sources provide a surprisingly detailed field guide dedicated to distinguishing the slimy spike from its closest lookalikes.
Yeah.
Let's run through the lineup of these forest imposters, because there are several, and they share that same bizarre biology.
Sure. The primary confusion for foragers happens right within its own genus. The most common lookalike is Gomphidius smithii. At a glance, standing over it in the forest, it looks remarkably similar. It has the slime, it has the decurrent gills, but structurally, it's generally a smaller, more delicate mushroom. However, the definitive key differentiator is found at the stem base. While our slimy spike has that unmistakable bright chrome yellow base, G. smithii tends to have a pale pinkish or sometimes a faint wine-colored tint to the lower stem.
Okay, that's a clear visual cue. Yellow base means glutinosus, pink base means smithii. What about Gomphidius organensis? The name suggests it's a Pacific Northwest native.
It is, and organensis is notoriously difficult to tell apart from the slimy spike in the field. It shares the exact same spruce habitat, the same slimy exterior, and the same general grayish-purple coloration.
So how do you tell them apart?
It tends to exhibit a different growth pattern. It usually grows in tight, overlapping clusters, what mycologists call a cespitose growth habit, whereas glutinosus usually grows singly or widely scattered across the moss. But to be absolutely scientifically certain between the two, you must rely on a microscope.
Spore size again.
Exactly. The spores of organensis are significantly smaller, typically only measuring 10 to 13 micrometers long, compared to the massive 15 to 21 micrometer subpuciform spores of the true slimy spike.
What if I'm foraging in a slightly different environment? Like, what if I find a slimy, decurrent gilled mushroom growing exclusively under a larch tree instead of a spruce?
If you were explicitly under a larch tree, you are almost certainly looking at Gomphidius maculatus. This species is a highly dedicated specialist. Its underground network only associates with larch.
Does it look different?
It differs physically because it lacks the thick, universal glutinous veil of our main subject, usually only having a sticky cap, and its flesh has a very distinct, rapid oxidation reaction. If you press your thumb firmly against the gills, they immediately flush a bruised wine red before eventually spotting brown and blackening as they age.
And finally, there's a cousin mentioned in the sources that isn't even technically in the Gomphidius genus, but looks enough like it to cause confusion.
Right, that would be Crogomphis rudilus, commonly known by foragers as the copper spike. It belongs to a sister genus. First, it prefers to grow under pine trees rather than spruce.
Right.
Visually, instead of a cool grayish-purple cap, it sports a very distinct warm coppery or reddish-brown cap.
Oh, that sounds pretty.
It is, and the ultimate tell is the interior flesh. If you cut a slimy spike in half, the flesh is stark white above the yellow base. If you cut a copper spike open, the flesh is a vibrant, deep orange ochre color all the way through the cap and stem.
It is fascinating how these incredibly specific ecological niches, spruce versus larch, solitary versus cluster, drive such subtle but vital physical differences in these organisms.
It's all about adaptation.
So to step back and summarize the incredible, highly improbable evolutionary journey of Gomphidius glutinosus, we are looking at an organism that looks for all the world like a standard agaric, complete with deep, waxy gills, but whose hidden DNA firmly places it in the family tree of the spongy boletes.
Yeah.
It presents itself above ground as a simple, friendly symbiont, helping spruce trees thrive. But beneath the soil, it's actually using highly evolved molecular pumps and haustorial syringes to ruthlessly hack into subterranean root networks and steal hard-earned food from Suillus mushrooms.
Precisely.
It protects its stolen loot with a bespoke chemical sunscreen called Gomphidic acid, a molecule which accidentally makes it the perfect, hyper-efficient biological vacuum cleaner for catastrophic nuclear fallout and toxic heavy metals. And if it doesn't happen to poison you with a concentrated dose of historical industrial waste, it tastes surprisingly like zesty lemon, provided you meticulously peel the slime off before it oxidizes and turns your entire foraging basket pitch black.
It is a masterclass in biological contradiction. And
Yeah.
to you, the learner listening to this, I think the most important takeaway from this deep dive is that the natural world is infinitely more complex, nuanced, and interconnected than the simplistic labels we desperately try to apply to it.
So true.
We draw these neat, comforting boxes in our textbooks: parasite, mutualist, guild, pore, toxic, edible, plant, fungus. But Gomphidius glutinosus walks right across every single one of those boundaries with absolute impunity. It is a highly hyper-specialized organism, perfectly adapted to its bizarre, extreme niche, and its very existence constantly challenges our fundamental assumptions about how ecosystems function.
And it leaves us with one final, truly provocative thought to mull over. We started this deep dive by talking about X-rays, precision, and finding ways to clean up our worst environmental disasters. We've just spent an hour learning that Gomphidius uses haustoria, those microscopic biological syringes, to actively selectively hack into a specific fungal network, stealing resources without crashing the overall forest system.
Right.
And simultaneously, we've learned that human scientists are proposing that we use Gomphidius to clean up our worst nuclear disasters by sucking radioactive cesium out of the soil.
The intersection of those two seemingly disparate biological facts is exactly where the future of environmental science lies.
Exactly, because if fungi have evolved the innate biochemical ability to so elegantly hack each other's underground networks
Right.
to survive extreme environments, what happens when human scientists finally learn to fully decode, control, and program that haustorial hacking mechanism?
Botox abilities.
If we can understand the precise molecular language that allows the slimy spike to tap the wire, could we one day actively program fungi? Could we engineer a bespoke fungal internet where specific designer mycelial networks are dispatched to target entirely new man-made pollutants like microplastics or forever chemicals, or even actively cure diseased forest ecosystems simply by rewriting the biological code of the soil?
Here's a huge question.
The slimy spike isn't just a bizarre slimy curiosity in the woods. It might actually be the literal blueprint for the next century of advanced bioremediation.
It forces us to look down at the dirt of the forest floor and realize that we are standing on top of an unfathomably complex biological supercomputer. We are only just beginning to learn how to log in. Thank you for joining us on this exploration.
Thank you for coming on this deep dive with us. Keep questioning those neat little boxes, look closer at the world around you, and we'll see you next time.